Good morning. I'm waiting. We're nearing the end of the academic year, aren't we? As we near the end of the academic year, the graduating fellows are stacking up all of their grand rounds in case anybody hasn't noticed. And today we have Billy Chen, who is graduating from our fellowship in June. Billy came to us from Boston, but he has an interesting past. He was a graduate of Cal State Los Angeles, where he majored in biology, but he had a minor in mathematics, a minor in microbiology, and a minor in theater arts, which is very interesting.
How old was Billy when he graduated?
12?
Billy, how old were you when you graduated?
I was 17 at the time.
17?
Yes.
When he graduated, yeah.
When he graduated. But I think the theater arts, I mean, we can all agree that Billy looks cheerful under incredible stress, no matter what service he's on. And I have this suspicion that the theater arts may have given him the skills to do that. So anyway, Billy, at a young age, went to Boston University to get his MD, PhD, and then went on to residency at BU. Came here to do to work, kind of find himself on the West Coast again, I would say. Worked as a hospitalist on the MICU services, and at that time joined Chuck Murry's lab and did essentially our research first pathway with 2 years of research in the lab, followed by his 2 clinical years, which he'll be following, finishing up, as I said, in June. And in Chuck's lab, he became an interventional primatologist, and did accomplish amazing work, which has resulted in a Nature publication, which he's going to describe today. So here's Dr. Chen.
All right. Thank you, everybody. I want to first take an opportunity to thank the division for giving me the opportunity to present some of the data that and some of the work I did in Chuck Murry's lab before starting my clinical training. So this is very exciting for me. So the title of the talk is Unbreaking the Heart, Cardiac Remuscularization in Non-Human Primate Models. I have no disclosures. I'm a poor fellow. So the overview of the talk. So we're going to go into and describe briefly the history of regenerative biology, go into a little detail about the history of stem cells, and prior efforts in the field of cardiac regeneration. And after that, we're going to kind of get into the meat of some of the data that's come out of the lab, some of the work that I've done in the lab, as well as some of the limitations of the studies before we finish. So to start off, and I always feel a little silly kind of bringing up, especially in this audience, this slide, so talking a little bit about ischemic heart disease. So we have, a coronary artery that supplies blood to the myocardium, and in the event of, of ACS or occlusion of the vessel, you decrease the flow to the myocardium, resulting in ischemic injury to the myocytes, to the point where you'll then have subsequent cell death and injury to the heart. Now, thanks to guideline-directed therapies, as well as our advances in revascularization, a lot of that actually improves, and we see a lot of cardiac stabilization. But even with stabilization, you still see that there can be progression from both the injured heart as well as the recovered heart toward remodeling and cardiomyopathy. Now, the heart in itself is one of the least regenerative organs in the body. It essentially heals, as it were, by scarring itself, so leaving regions of the heart with areas of fibrotic tissue that is non-contractile, that doesn't really function. And from there, it can progress, like we were saying, to heart failure. And once a person is diagnosed with heart failure, you're looking at a 50% survival in 5 years. So that kind of sets the stage for kind of the proposition that we're making. The question that we're asking really is, especially in the era of stem cell biology, cell therapeutics, can we use stem cells as a building block or products derived from stem cells to remuscularize or repair the heart to prevent or reduce heart failure? Now, to talk about that, I'm going to take you on a little bit of a history lesson. So the idea of regeneration has been around for essentially as long as people have been observing animals in the wild. You know, there's regeneration occurring naturally in all over the place, but it wasn't really formally described until the 18th century during the Age of Enlightenment by folks such as René Antoine Ramier, Abraham Tremblay, and Lazzaro Spallanzani, all of which were more observing these phenomena, these instances of regeneration in their respective organisms of interest. In the case of Ramier, he was looking at crayfish limb regeneration over here. And then for Tremblay, he was looking at regeneration of the hydra, which, you know, for his purposes, he was trying to figure out whether or not it was plant or animal. And then the model that a lot of us are more familiar with growing up, the idea of newts and salamanders being able to regenerate limbs when they're removed. Unfortunately, that resulted in a spate of headless and limbless salamanders in this countryside of Italy for a while. But this goes on to describe that, you know, a lot of this original thought happened way back, even just observing animals. Now, the field of regeneration, or the thought of regeneration, kind of stalled, you know, around that time. You know, they didn't have the tools of developmental biology, embryology, or anything moving forward. So it kind of stalled, and then it didn't really kind of progress more until the 1900s, where we had the advent of these respective fields. So that brings us to the next topic. What is a stem cell? So fast forward into the 60s. So in 1960s, Ernest Macaque and James Till identified what is now known to be hematopoietic stem cells. They were looking at irradiated mice and saw that there were colonies of cells that are still growing and proliferating in the spleen. Now, from their discovery, even though they didn't discover what we now call pluripotent stem cells, they were the first ones to kind of formalize the definition of what a stem cell was. Now, it requires a cell to be what we now call undifferentiated, but still have a high proliferative capacity, meaning that the cells are still able to divide. But with that, they're able to give rise to more specialized cells later on, or in their daughter cells, so they're able to further differentiate. Now, some of the milestones after that, continued. So in 1978, stem cells were also discovered in humans. But what really ultimately kind of lit the spark in the field was kind of the ability to generate stable and cell lines for stem cells. And that happened in 1981 when they first developed the mouse stem cell cell line. And we really didn't get to humans until about 1998, when the first human embryonic stem cell lines were generated. Now, it's around the same time that the field of cardiac regeneration kind of also burgeoned. And some of you may know who this is. This is Piero Inversa, and he's been in the news as of last couple of years, unfortunately due to some of his later work having to be retracted. But about 1998, he was one of the first few to kind of put out that the heart was not as, unregenerable. No. So the heart still had regenerative capacity that was contrary to kind of the popular belief at the time. And it was from that initial paper that kind of sparked a slew of other investigators to look toward other sources of potential cells that could be used for repairing the heart as well as other organs. So at least in the field of cardiac regeneration, folks turned toward sources such as the bone marrow, where they were able to isolate progenitor cells such as where we already knew about the hematopoietic stem cells, but other cells like endothelial progenitor cells, and other bone marrow-derived cells that had the potential to differentiate into other cell types as well. Afterwards, people started looking toward more somatic tissue types, such as muscle, adipose tissue, where they're able to isolate what we now call mesenchymal stem cells. Then folks also turned their attention back to the heart itself. Did the heart have regenerative potential like Inversa had postulated in 1998, and where they were able to derive what they believe to be cardiac stem cells and cardiospheres, which are kind of organoids or bundles of cells derived from these stem cells. Now, discovery of these cell lines kind of started a whole series of trials that began in the 2000s. The trials were a little mixed in terms of their results. Like, the results ranged in many of these studies from these therapies or these cells doing nothing at all, to some of more positive results, one of which was the Poseidon study, which looked at these bone marrow cells and showed that there was subjective improvement in patient functioning, but no other measurable changes. Scipio, which was, where is Scipio? Up here, which was one of the studies that directly came from the isolation of cardiac stem cells, saw that there was a reduction in infarct size. And Caduceus, which is the one that utilized the cardiospheres, demonstrated increase in viability of the heart tissue as well, but they did not show that there was any change to function such as LVEF. Now, regardless of the cell type, there was a couple of mechanisms of which they thought these cells were benefiting the heart or improving the heart function in the studies that were positive. One of which was angiogenesis, new vascularization, where they felt like when you injected the cells, they would recruit, they themselves would differentiate into potential new blood vessels or recruit endogenous or cells that are already in the tissue to form new blood vessels. A more widely accepted or a more widely thought of mechanism was this paracurrent effect that a lot of folks talk about, and you may recall Dr. Bonwell talking about when he was here lecturing. And in essence, it does a lot of these things where the cell is injected. They don't necessarily engraft or sustain, but by being there, they stimulate a lot of these mechanisms such as matrix remodeling to kind of alter and attenuate fibrosis and scarring, modulate inflammation by sending signals to surrounding tissue, and regulate cell differentiation, including also neovascularization. Now, the last mechanism, I think, is where we start getting into our neck of the woods, is this idea of cardiogenesis or remuscularization, using the cells as a building block to make new cardiac myocytes in the heart. Now, I guess the difficult is not the right word, but a lot of these cell lines that we've been talking about are what's been considered adult stem cells. Now, adult stem cells from these studies have not entirely been shown to really engraft that well, and thus the mechanism of the paracurrent effect was so dominant. But around the same time, there were labs are also using pluripotent stem cells, such as human embryonic stem cells, induced pluripotent cells as well, where they showed a better engraftment rate, where like they were able to show that the cells would be injected into living organisms and they would stick around and survive. Now, embryonic stem cells. So I always thought of embryonic stem cells much like fellows. If you don't give them any sort of guidance or any control, they kind of just run amok, right? So this is one early study where they injected murine endocardial mouse embryonic stem cells, and it formed a teratoma. So, the cells were able to differentiate into all three layers of the germ line and then produce tissue that we weren't necessarily hoping for. But here at University of Washington, we have folks who have the expertise that were able, who have been able to direct these pluripotent stem cells into what they wanted, especially in our labs over at South Lake Union, Chuck Murry's lab. They've been able to help coax these cells along through small molecule differentiation pathways into developing into cardiac myocytes. Oops. Okay. So with these myocytes and these building blocks, and kind of going back to the original question, can we use these cell therapies or these building blocks to heal the heart, to rebuild the heart? So, in the following studies, we're hoping to show that we can use these human embryonic stem cell-derived cardiac myocytes to remuscularize the infarcted heart model. And in our case, we're going to be using non-human primates. And we also can show that these cells are able to mature in the animal itself, as well as integrate into the host heart that we inject them into. And lastly, that the remuscularization actually leads to some sort of improvement in function. And in our case, we're looking at the LVEF. So we've broken this large multi-year spanning project into a couple of phases. And the first phase Chuck has described always has been the kind of phase one, looking to see if the ability, like pilot study, to see if this is even feasible. Now, previously in the lab, folks have been able to show engraftment of these embryonic stem cell-derived myocytes in small rodent models. But we wanted to take it into a larger model, and in this case, we're using the macaque because we felt that it was a better approximation of human disease. Now, the model itself requires a timeline of how the animals are processed. So day 0 really is the day that we inject either cells or control. So 2 weeks prior, we induce a MI, leading to scar formation, leading to injury. And then on day 0, we inject the cells, and then at a later endpoint, either 2 weeks out or 3 months out, we collect the tissue, we look at how the cells are doing to see if it's really doing anything. So these cells are delivered in a way where we are using a thoracotomy method. So we really wanted to be able to kind of see the infarct that we're injecting, have direct visualization, and know exactly what we're popping these cells into. So the cells, we're injecting about 750 million myocytes that were grown in the lab through 5 different injection sites each time. You can see, I don't know if you can see these tiny little sutures here. Between sutures injected and tied off in order to make sure that these cells are staying within the tissue that we injected. And from the pilot study, from the initial study, we're able to see that yes, the cells do stay within the tissue. They do grow. So this first set of images is looking at a slice of the heart through a short axis. So this is your short axis view. The red is staining for both human and monkey tissue. Now the green is staining for human tissue specifically. These cells at the time were made to express a molecule that flashes and glows green with each contraction. So they have a green fluorescent protein moiety on there, so they were able to probe for that and show that these large swaths of green tissue are human tissue. So in both in A, B, and C, you're able to demonstrate these large areas of human myocyte tissue. In subsequent images here, they are looking at tissue that's collected now 3 months after they initially injected the cells. Now, what we're staining for here, and the real take-home is really these 2 images, is that within the midst of all this green tissue, which we're presuming to be to be human graft, that they're developing these rings of red, which are staining for endothelial cells, or kind of the building blocks for blood vessels. So from that, we were able to show, or at least see, that the graft itself is becoming vascularized, where in essence, when we inject it, it's just a slurry of cells, but now it's organizing itself into what we would call cardiac myocytes, as well as developing these blood vessels. Now, these are two rows for comparison. One is at 14 days, one is again at 3 months. And this is to look at the maturation of these cells inside the animals that we've injected them in. So after 2 weeks, we're staying here for actinin, which is basically one of the proteins found in the sarcomeres of these cells. So if you ever looked at heart tissue under a microscope, you'll see these nice linearities of the sarcomere. So between 14 to 84 days, you'll see that there's increased content, or increased amount of these of this organization of these myofibrils. And also, it's hard to appreciate on this projection, but there's also an increase in connexin 43, and that's a protein that makes up a gap junction. And this is going all the way back to high school, college biology, where the gap junctions are these connections between the cells that allow the cell to share information back and forth. And kind of one more set of findings to demonstrate maturation of these cells. So here, again, this is the top row is tissue obtained from animals after 14 days. And the bottom row is tissues obtained from animals 3 months after injection of these cells. So you'll have yellow, which is what we consider the edge of the graft, meaning that it's right at the interface between host, meaning monkey tissue, as well as human tissue, or core, which is just smack dab in the middle of the graft. And again, we see kind of increase in alpha-actinin staining, showing that there's more organization over time between 14 and 84 days. But also, we're able to see that there's increase in NCadherence, another marker, another protein that is seen in intercalated discs. So between each of the cells or of the myocytes, intercalated discs and NCadherin allow for both transmission of mechanical stress as well as electrical signals between the cells. Now, if this will play, which it may not, okay. So the hope was to show you this wonderful video that the lab was able to generate. Basically, you know, we have one of the hearts that has been injected with the cells, and you're able to see these regions of flashing, well, in this case, black and white, but these regions of flashing that correspond to where we injected the cells. And this is some of the data obtained from that sort of preparation, where the blue and red tracings are the flashing, the observations of the glowing from these two regions of observation, and we're corresponding them to the heart's native conduction. So from here you can see that from two different places on the heart that they're beating in sync, and also in this case they're beating in sync with the animal's native rhythm. Now, to make sure that that isn't just coincidence, the lab also paced these cells, atrially paced the cells, and these grafts are in the ventricle, so they were again able to show that these separate grafts were able to beat in sync, but also with the rate of the pacing. So these grafts were at least electrically coupled from this observation. Now, the data was later kind of put together and published in 2014, and the takeaway from these set of experiments was that the human embryonic stem cell derived cardiomyocytes can engraft and mature in the host heart. Interesting enough, when we did this experiment, or when the lab did this experiment, an unexpected finding kind of popped up, and it was that the animals would develop or had developed these non-fatal ventricular arrhythmias that weren't observed in smaller animals. And the thought at the time was that because the baseline heart rate for the macaques are so much slower, that whatever rhythm that these cells were generating is now more evident, where in the smaller animals, their baseline heart rate's like 200 beats per minute, 400 beats per minute, depending on the size. You wouldn't really see that tachycardia, and it was more evident here in these larger animals. And at the same time, there was no significant change in systolic function. Now, why was that? We were able to demonstrate that it is integrated into the heart, and it contracts, and it's electrically coupled. So why didn't we see any change in systolic function? And part of that, we think, was due to how the animal was prepared. And what I mean by that is when we cause the infarct, you know, what we do is we put down a balloon and occlude the coronary artery for about 90 minutes. And then the occlusion was really distal, usually distal LAD. So the infarcts would be relatively small, and you'd have a reduction of EF from baseline 65% to about 60%. So really only a 5% drop in observed decrease after giving the animal model an MI. So to better assess whether or not this, you know, this therapy works, we wanted to give ourselves a wider therapeutic window. So we developed a timeline where we're now causing larger heart attacks or larger MIs in these animal models, and instead of just looking at it 2 weeks to 4 weeks, now we're taking these animals all the way out to 3 months again, but we're also including EP studies to help better explain potential mechanism for these VTs that we observed, as well as doing cardiac MRIs to better tissue describe the tissue that we're seeing, as well as a more accurate assessment of heart function. So for this phase of the study, we enrolled 14 animals, of which in the end we only were able to analyze with an MRI 9 animals, 5 of which were assigned to a control, 11 were assigned to treatment. Many of animals which fell out for complications from the procedure, not necessarily from cell injection, but just even the process of creating the infarct. So this is an example of the infarcts that we created in the second phase of the study, where you see this is the Macaque LAD, their baseline ECG. Then we go and include for 3 hours in the mid to proximal LAD, leading to what we can typically see with STEMI, these large tombstone ST elevations suggestive of large infarcts. Now, histologically, from these large infarcts, now we're able to see from these large transmural regions of scar. So this is another cross-sectional slide of the left ventricle, where the green is stained for healthy myocardium, and the pink is basically connective tissue and scarred down myocardium. Now, part of the difficulty in the second phase of the study was because in the first phase, the cells that we're injecting was made to express this GFP molecule. And in the second phase, we're now using cells that were quote unquote cleaner. We're trying to minimize the number of foreign epitopes that it was expressing. So it became harder for us to identify these cells in the histology. So we had to come up with new ways to identify the tissue. And after prolonged search, we were able to find these particular antibodies for us to visualize the human grafts. So again, a section of the short this is the kind of the scar portion of the short axis of one of the hearts that we infarcted. So pink or magenta here is staining for healthy myocardium. Blue is all scar. So here you see kind of the thin wall, some patchiness, and some healthy myocardium, but mostly these long areas of blue scarred tissue. Now, after injection, and we're looking now at the animal's tissues a month out, now we're able to demonstrate these large, again, patches of new growth or new human myocardium. So this is consistent with the prior phase, where now we're able to demonstrate again that you have these cells regrowing in these areas of the heart that we're injecting them in. Now, rather than going through all the similar steps of validating maturation, we're able to find able to do a couple of new things with this study. One of which was to look at the phenotype of the cells that we're expressing. So these are representative images from both 1 month after we injected the cells and 3 months after we injected the cells. And we're looking at two markers. One is myosin light chain 2A, one is 2V. The 2A is a marker for more immature cell types, including atrial, nodal, as well as mature ventricular cells, where the 2V is demonstrating more mature ventricular myocyte phenotypes. And at 1 month, you can see that in the graft here, the host, you could see that it's already staining for the mature phenotype. In the graft, you have this kind of lacy cell structure with a lot of, well, not a lot, with more grain, demonstrating that it's still relatively immature as grafts go. But after 3 months, or 2 months in addition, again, you see this better organization of these cells in the graft and much less of a green, suggesting that there is a conversion of phenotype. There's a change or less expression of immature markers in the cells. Similarly, we looked at, again, from 1 month to 3 months, looking at another marker of kind of immaturity. This stands for less mature form of troponin, as well as the red staining for human troponin, which is more abundant in more mature cell types. So again, from 1 month to 3 month, more red, less green, and in this third panel, we're staining for looking at the T tubules that are expressed in between the sarcomeres that allow the cells to contract. Another sign or feature of a mature cell. So I won't belabor the point, but similarly to what we looked at in the previous phase, we looked at connexin and cadherin in demonstrating between 1 month and 3 months that while you do have expression at the 1 month period, they're much more organized in the later times, more toward where we imagine to be kind of in between at the end caps of these cells, in between the cells where the connections need to be. Now this is probably one of the coolest parts that came out of the study. So one of the things we were able to see is that yes, the graft grows in host tissue, but we haven't been able to clearly identify until this portion of the study that actually integrates and connects with the host tissue. So in white is where are cells from the monkey heart, and then the green again is stating for markers of the cell-to-cell connection. And here you can see troponin in the red part staining for the human graft, the white being the monkey host cell, and this portion, this intercalated disc right here, showing the connection between the two. Similarly, we're able to also demonstrate staining for CD31, that marker for endothelial cells, that there again is vascularization in the midst of the graft. So as we had described at the beginning of this portion of the study, we're also using cardiac MRI in order to visualize both the tissue itself and also to better characterize the function of the heart, looking at the EF. But one of the cool things I think about the study was that we were able to actually visualize some of the graphs by MRI itself. So before at baseline, this is looking at a short axis where the black is the myocardium of the left ventricle, and this kind of dulled out area is a region of scar. But then after 1 month, our radiologist basically came back and said, what are these things, or could this possibly be new myocardium? And at 3 months, these little nuggets that weren't before here at baseline were still present and got us pretty excited. So we were able to take that animal, or at least a couple of the animals, and get the tissue stain for in the regions where we think these correspond to in the imaging, and demonstrate that there's these large swaths again of new grown host tissue to the point, and they're large enough where we're talking about regions about centimeters wide or long that were now visible to be seen on the MRI. Now, from the MRI data, so again, just to kind of back step for a moment here. So the study is relatively small. We did have 17 animals enrolled. In the end, we only had 5 treated animals and 4 control animals. So the data isn't conclusive, but is very suggestive of a few things. One of which is a change in EF, and we'll talk about that right now. So at baseline, we're looking at the EF of these animals, and looking at the infarct size of these animals at baseline. So this is after infarction, and over time, the animals that are treated with the cells have a reduction in the size of the region of infarct, as seen in the MRI, where the control animals who received vehicle didn't really have much change, and this is better demonstrated here on this graph, where the difference in the size of infarct between baseline and 4 weeks after is markedly different. So looking more at the function of the heart, so the baseline LVEF of macaques are pretty similar to humans. Greater than 55%. Majority of our animals actually were in the range of 65% in terms of their LV function. After their infarct, and again, this is our goal, was to demonstrate a large reduction in their EF. We were able to decrease their EFs by 20-some points. The average ejection fraction after the infarct was about 45%. Now, 4 weeks after, in these 9 animals, we were able to demonstrate that at least in the treated cell animals, that there was a trend for improvement in LVEF, while the EF for the control animals stay relatively same. There is some recovery as expected in untreated patients. So to better visualize that, again, if we looked at the change in LV function from baseline to 4 weeks after, there was a much greater change in the subjects that were treated with the stem cells. Now, in terms of absolute improvement of LVEF, we're looking at about 8% difference between the two groups, which I think, taken in totality, I mean, it is an improvement. It may seem modest, but I think compared to some of the other therapies we have out there, it is actually one of the better treatments in terms of improving EF if this were to bleed. Now, the cool part about this is that from a couple of animals that we took out even further, we believe that the function continues to improve as the animals go out further. So again, this is the same graph that we showed just now. So you have an improvement, drop from the EF into the 40s, and then improvement up to maybe about 50%. But in 3 of our animals, similarly, EFs drop down to below 45% with improvement after a month. But then looking forward at 2 months and 3 months, we're able to continue to see improvement in their EFs, almost to the point where at least one animal almost appears like their EF is back to baseline, where you see the decline of the untreated animal continue over time, as we expect in our heart failure patients, to see at least initial some recovery and then continued decline in their function. So in terms of take-home points, again, in terms of what we were able to show in our studies, in both phases of the studies, we're able to demonstrate that these cell therapies, or at least the derived cardiac myocytes, were able to remuscularize regions of the heart of these infarcted non-human primates. We're also able to demonstrate that they also mature and integrate, especially in that second part. We're able to see them really kind of weave and interknit with the host myocytes. And lastly, the remuscularization leads to improvement in left ventricular systolic function, although this may be a little harder to elaborate on, since we're looking at a little smaller and with fewer animals going out toward 3 months. So that brings me to the limitations of the study. So the initial question that we proposed was, can stem cells or stem cell-derived therapies be used to remuscularize or repair the heart and prevent or reduce heart failure? Now, this was kind of a sleight of hand, because the question asks is ability to improve heart failure, where a lot of our models thus far has been looking at kind of acute, subacute MI models. Now, I know there's been attempts to develop a more heart failure-like model, but in terms of at least the time that I was in the lab, the logistics of it was just a bear in terms of being able to create these models, have them go out long enough clinically to actually have clinical symptoms of heart failure, and keeping them alive at the same time was just really difficult. EF as a surrogate for the outcome. So again, we're measuring primarily looking at the EF rather than what a lot of us look at in terms of therapies that we give our patients is how does it reduce mortality or morbidity. But in this case, because we had a hard endpoint in terms of collecting tissue from our animals, EF was the surrogate that we looked at to see if there was improvement in function. And there's literature out there that shows that EF is a good marker for mortality in our patients as well post-MI. But one potential is to see maybe in the future to carry out an experiment to a clinical endpoint as opposed to a planned one. Now, again, the second phase of the study is limited by a small number of subjects. From this diagram, a lot of the animals were excluded primarily for reasons outside of either the cell injection but primarily from the infarcts and other procedures performed during the study. And there's an issue of immunosuppression. So, we didn't talk much in terms of immunosuppression, but in terms of making this a potential valid therapy moving forward, the immunosuppression, at least in the get-go, we just wanted to see something work. And we kind of brought up the big guns, and then over time, looking toward the future, we still need to look at how to best minimize or tailor that therapy. So we're not just using large amounts of immunosuppression. The last point, which I actually will leave somewhat open-ended because I think this will be addressed in future talks, is this question about the arrhythmias that we saw. And I don't think the video is going to play again. But from at least our initial EP studies in the animals that did develop VT, it looked like, or ventricular arrhythmias, it looked like it was a focal source that corresponded primarily with regions where we injected the cells. It didn't seem to be, at least at the time, to be due to any circuits or mediated tachycardia, and it was primarily ectopic from a focal source. So with that, I'm going to conclude my talk. I want to thank the Murry Lab for all their support and time over the years that I've been there. The folks here at UW, Primate Center here at UW, for helping us see these animals through, and as well as want to thank everybody in the fellowship and the folks that I've worked with on the floors as well. Thank you.
So Billy, you know what my question is, fibrosis. Have you quantified the amount of fibrosis in the hearts? Is there a difference in the total fibrosis when you inject the stem cells? Or when you treat the primate with the stem cell versus the control?
Right, so in the studies we have used the imaging from the MRIs to kind of quantify the amount of fibrosis, the amount of scar. So in terms of specifically fibrosis, might be a little more difficult to answer, but in terms of if we're just using scar as an approximation of fibrosis, we were able to quantify, and it does look like it gets less with treatment with the cells.
So is that a total scar burden versus percent scar versus?
It's percent scar, so we were looking at how much of the left ventricular mass was scar at the time.
Okay, but you haven't looked at distant uninjured myocardium for
Not that I recall. Not that I recall.
Rick.
Nice talk. I have an echo question, which is probably the only kind of question I can have on top of this. But did you look at different ways of quantifying EF by echo, or did you use a standard method? And second, did the echo readers know which arm the
That's a great question. So the first phase was by echo, primarily the function was assessed by echo. The second phase was actually primarily done by MRI. Due to the difficulty of actually quantifying, or the perceived difficulty of quantifying and doing echo studies on the animals, because their heart orientation is actually very different from ours.
So the EFs you showed were the MRIs.
Correct. And the people who reviewed and analyzed the cardiac MR data were blinded to the animals.
Thanks.
Fareed.
Really fantastic talk. Obviously, lots of great work. I wanted to ask you about, you show maturation. Do you know anything about the number of cells? I mean, are they proliferative? Because maybe there's a trade-off where they're immature, you could have arrhythmias and issues, but since they're mature, they're proliferating, you have more cells. I know it's a small study. Do you have any thoughts about, do you think the number of cells are the same, they're more?
Sure, I mean, that's a great question, and the honest answer is I don't know. What we believe to be happening is that after we inject the cells, there is still a kind of a huge die-off of cells. When we inject it, not all the slurry that we inject is going to live, and in essence, there may or may not be a pericurrent effect still from the dying off of these cells, but the cells have a more immature phenotype, as you saw when we first injected them. There is what we think is a period of proliferation where then they develop and grow into these kind of large swaths of graft.
David.
Yeah, it's a very exciting technology. I have a couple of questions. One is, can you differentiate whether the immature cells are dying off, leaving you with a higher percentage of mature cells, or whether the immature cells are maturing?
That is a good question. I don't know if I can answer that. I don't think we've had a way to differentiate that specifically, meaning that to track specific cell phenotypes through time. So we can, with the histology, we can only kind of get these cuts and snapshots. So to be able to follow immature populations of cells forward, I think we haven't looked at.
The other question, you're clearly doing the studies that need to be done at the beginning, just to find out whether there's some type of efficacy. Ultimately, this would presumably be an add-on to guideline-directed therapy. So there must be a plan in the works.
Yeah, so I mean, a lot of this is me kind of, you know, holding the cards close to the chest because next week is Kenta's talk. And a lot of this, it may or may not be covered in what he's talking about, but there has been thoughts moving, at least by the time that I was moving back into clinical training, about using guideline-directed therapies in addition to this to see if there's that delta to be seen. But that can be addressed later in future talks.
Anyone else? All right. Thank you, Billy.
All right. Thank you, everyone.